our debate. We will have about 15 minutes. I would like to invite Dr. Tarcisio to lead the debate. Good morning to all. I would like to thank the organization and William for the invitation, for the privilege of participating in this seminar, which is so productive and is conveying so many interesting information of all origins. Starting the debate, I would like to use my title as coordinator of the debate and I would start asking Dr. Anna and also to Dr. David. The question is, considering parameters for toxicity, especially for lung toxicity, we have used classically in toxicology the mass with per air volume, milligrams per cubic meter. In nanoparticles, the mass is too little, so I have to imagine the volume of carbon nanotubes that are 5 milligrams per cubic meters, which was used in the exposure to rats in the case of nanotubes of Professor Anna. I would like to ask if it's not time to work in the development of a parameter that combines the square meter per gram issue and reactivity in the same size, to work this size, combining it like we do in general technology. I don't know if you have understood this question. Maybe I should reformulate it.
If you will correct me if I'm understood not. Yeah. Okay, let me just take it out. Otherwise, it's like mixed up. What language should I talk? Maybe Russian. Yeah, you're talking about certainly very important issue regarding the characterization of the particles, and I'm so glad David was devoted part of his talk regarding the various surface reactive properties of the materials. But when we're talking about carbon nanotube, keep in mind it's very highly aggregatable materials. Certainly we did it all possible characterization of carbon nanotubes, surface area. We did it like, some zeta potential. But when you use exposure, this is agglomerated materials exactly. Yeah, but what I would like to let you know that it's surface area of, not surface area, it's how it's called, BET parameter of aggregated of inhalation study was around 1,000. But if you like in a pharyngeal aspiration, 1,000, so it's more have surface area, and then when we have partially clean these materials. But when we use inhalation exposure, it was materials was used in the industry. The certain surface area is much lower. It is around measurement by BET parameter around 600. But if you compare the toxicity of this effect, because it's rather difficult to talk about when you deal with in vitro assessment and then realistic in vivo. In vivo exposure, inhalation exposure, give you more deposition of particle in the lung and give you the concentration. If we compare particularly these dirty materials using pharyngeal aspirations and inhalation exposure, and the dose which we deposit 5 microgram per meter cube was equivalent to the 10 microgram which you give the same material by pharyngeal aspirations because of the deposition of these materials. And you're absolutely right. When we have like for high put through materials assessment, it's very important to take to consideration charge of the particles and surface area reactivity and some like catalytic metal which we have been using. But keep in mind when we dealing with real situation in an inhalation system, it's a little bit different.
Queria agradecer, queria ouvir.
I would like to thank you and like to listen to Dr. David.
Certainly something that's going to be very important over the next few years. Here's how I see it. Essentially, we have years and years of measuring gravimetrically milligrams per meter cube, but as you mentioned, sometimes you have to measure all day to even get a sample, and you're not sure if that's an exact sample. So we have to go to other dose metrics such as particle number and surface area for things like setting occupational exposure limits or determining what workers are being exposed to. The problem with that is that A, we don't have a history of those sorts of data. B, what I'm being told by the exposure assessment people is that background is a real problem, because if you've got a truck outside, or if there are a lot of people jumping around or talking, you could have 10 to 15,000 particles per cc just as your ambient background, and that can change. And so to get essentially an accurate assessment of the particle number, and even after you have that, you don't know what it means. If I have 100,000 particles per cc, what does that mean? We recently published a paper 6 months ago where we did freshly generated amorphous silica fumes and measured particle numbers, but we actually back calculated to try and get a gravimetric assessment. Then when you talk about surface area, what if you have mixed particle types in different surface areas? So I think that my simplistic opinion is that we need to measure for the next 5 or 10 years all of those and try and get some kind of sense out of what is a reasonable amount. Once also you get into things like nanofibers, you've got aspect ratio, which is length and diameter. So you have different metrics. You're measuring fiber numbers per cc or per mil. So I think it is kind of a mess, but we need to get started to try and make some sense out of it.
Queria agradecer.
I would like to thank Professor David. And after asking my question, I would like to open the floor to people who want to ask questions, please. David. I think we're just starting. We're starting with like the end of the American Congress of Toxicology. Are we going to measure it all, or is it not necessary to measure? That was your discussion with Nigel Walter. We have to make many metrics or not? That was the final question, and you said there was a need for several metrics for the next 2 years. My question is about that, more or less, for you and for Anna. In your product, when you show a few toxic effects, the products were essentially of titanium oxide of rutyl. And rutyl is compared the product, amorphous product of anatase, and to the products of rutyl. And rutyl is less toxic. It has on other crystal phases. I want to know if this product is a basic rutyl product to be less toxic, because the amorph product is much more toxic in nanoscale. The titanium dioxide as routine. So I ask if it's an essential product based in rutyl, or is it an example of the difference of the surface area and the crystalline form under the point of view of toxicity. Professor Anna, my question is not about the nanotubes of carbon of single wall, but maybe multiple wall. I read in the recent literature and the importance of the relationship between thickness and longness of the nanotubes. They're showing shorter nanotubes, leading to a process of chronic inflammation and larger nanotubes leading to phagocytosis, a frustrated phagocytosis, to a larger possibility of inflammation and maybe even problems in the pleura or induction of melitheliomes. I would like you to compare, to give your comments and doing some comparisons. I would like to know your remarks about that.
That's an excellent question, William, but it's actually more complicated than I even presented because, first of all, I talked about surface reactivity and crystal structure and pH, but actually when you take the P25, which is the UF3, it's actually to show that it's not all TiO2 is TiO2. It's designed to be a catalyst, and so not only does it have the features that I described, it doesn't have surface coatings, and it doesn't have a neutralized surface, and I believe, although I'm not a chemist, the reason is to catalyze reactions. The DuPont one is designed to go into plastics and paints, and its main function is UV scavenging and to provide a white or, in the case of the ultrafine, a less white pigment. So it has actually a totally different function. It is rutile, and actually the final product, which I talked about in the other studies, not the pulmonary, because as you're moving and developing a product, the final product actually was 85% rutile, 15% anatase, is the final product called DLS-210. But it has a totally different function than P25. And so its surface is very different, not only, and I think that's reflected in the surface reactivity. And another thing I mentioned, I didn't talk about, but what was on the screen was the pH of the P25 is very acidic. Which I think relates to its catalytic function. So I don't think it's just a matter of rutile versus anatase. In fact, the P25, although, again, I'm not a chemist, is 80-20, and maybe that was designed to be the best possible catalyzation product for catalyzing experiments, that particular ratio of 80-20, and not just 100% rutile, because actually the first set of studies that I talked about with the nano rods and the nano dots were all anatase, 100% anatase. So I think a lot depends on the chemistry of the particle, whether its surface is neutralized, whether it has surface coatings, and its surface reactivity, and all of those factors, and crystal structure. And I think all of those factors go into affecting what its application is going to be, and then also its toxicity. So I think it's not just a crystal structure, I think it's 5 or 6 different factors.
William, you actually brought a very great question, a couple of questions, if I understood correctly. Yeah, first of all, your question, whether we need to test everything. But let me just refer you to this new docket of EPA. It's carbon nanotubes, single wall and multi-wall, considered new materials. Its main manufacturer has to meet PMN, pre-manufacturer notes. Do we need to test it? If you would like to put through EPA, we do. And your second question regarding the more, I would say, safety and a kind of lyrical issue, like whether we can get mesothelioma, whether it's like short or long shaped nanotubes. First of all, I would like to a little bit correct you. There is differences between asbestos and the carbon nanotubes. Everybody knows that asbestos causes frustrated phagocytosis, because when the macrophages try to attack this material, try to engulf, they produce a lot of radicals. And the carbon nanotubes, we switch into totally different paradigm. Carbon nanotubes do not fully recognize like asbestos really foreign materials. They switch paradigm from a frustrated phagocytosis for production of cytokine. In particular, try to just like, save our body to make kind of inoculation of these informed granulomatous lesions there, It's mean it produce more type of TGF-beta and pro-collagenous cytokine. It's no frustrated phagocytosis. It doesn't mean it causes oxidative stress, but it's not by the mechanism of frustrated phagocytosis. And some other issue is very important, like you mentioned, like short and long-term carbon nanotubes. Certainly, short carbon nanotubes, but it's not compared to asbestos. But again, we're talking about different materials. Short carbon nanotubes doesn't produce much inflammatory response as well. Yeah, but the beauty of short carbon nanotubes, they just more easily can be taken to this mucociliary way to taken off the body. But some other new data, which is we published paper in Nature and Technology showing they can be degraded. Yeah, and can be degraded by virtue of some enzyme. Yeah, peroxidase, myeloperoxidase, and I have done some experiments with knockout myeloperoxidase mice. We just finished in the paper, just fresh from the oven. This mice has much less fibrosis. It's mean in this situation when animals can be given some like pro-inflammatory ingredient, you can just match fibrotic response. Yeah, and regarding the mesothelioma, there is only 2 papers published. One of them, it's P57 mice, which is Japanese people, which very high concentration of the carbon nanotube. It's caused like some type of cancerogenic outcome. But what we just discussed, I came from Nobel Forum, and then Donaldson and I discussed this issue, whether we can deal really with cancer. It's rather difficult to tell. First of all, how can it reach this high concentration in the lung? It's really hard. And it's rather difficult to have animal model to develop any cancer in the carbon nanotubes there, Yeah, and we have been using so far animal not prone to the cancer. What I would like to comment to you, what we try to get around this issue. I want to show you the picture, but a little bit maybe later. Yeah, you can inoculate mice with a cancer, which is in the lung, using Lewis cells. And in a normal condition, they develop some metastatic changes. But metastatic changes on the base of carbon nanotubes quadrupled. It's mean this is kind of indirect way showing it can cause cancer, but it's not specifically.
You mentioned the self-regulation of DuPont with the defense environment. This self-regulation was also asked yesterday. Dr. Rocco answered it, and I would like to know your opinion about the self-regulation issue, because this action of DuPont of environment defense had several entities, American entities and NGOs, that made a public manifestation, a public telling that this process should be public, and the regulations in nano should not made only by one company or by several companies, but in a public way. So I would like to know what you think about it. And to Ms. Zuleika, I would like to ask, I remember we were in MSCT 3 years ago, September 12, 2007. We wanted to create a bidding for nanotoxicology and economic social impacts for the cosmetic area, etc. At the end, this bidding did not take place. We followed the biddings, but so far there was no nanotechnological bidding for impacts, etc. So my question is, it seems that your work was very well done, and it was made within other projects that may be presented to another bidding, which are not mandatorily study-oriented, or none of them has demanded that you need to have a study for that. This makes me understand that this was your initiative to do this kind of study together with the project that was presented. This seems to be a feature that is made along this decade in the division of resources for nanotechnology. There are no resources for this kind of study, so I would like to know your appreciation. What's your name, please? Pedro. Just to take note, it is 5 to 11. We would like not to do questions after 11 because we would like to stop at 11. There is another person before. Okay, what's the name of that person? Just answer the question of Pedro first, and then what's his name?
Thank you for your question. From at least my perspective, DuPont has always had a strong tradition of product stewardship, and so I think that we realized it was important to try and develop a sort of risk management system, particularly with an NGO, in this case EDF, and it took 18 months, and I can tell you there was a lot of spirited discussions about how we should do that. But having said that, I think, frankly, personally, I think that's going to be a moot point because, and I think we will continue to do what we're doing but follow regulations. I think with all this effort now worldwide to define, which I think is very time-consuming, a definition of nanomaterials for regulatory purposes. And frankly, I personally, this isn't a DuPont position, wish that they'd spend more time generating good hazard data than all of these commissions all over the world setting a definition, because I think ISO had a pretty good definition. So I think it's all going to reach the regulatory arena very soon, and that companies developing new products with nanomaterials are going to be required to do at least hazard assessment, both environmental and human health hazard assessment work. I don't know how extensive that will be, but I think that's going to move into the regulatory arena very soon. But I think we will keep our number one, our base set, our nano-risk framework idea. We'll obviously comply with regulatory rules throughout the world. But, and I also want to make sure that you know that it's an evolving process. It's not like we set this and said, this is how it has to be. This is what we think is reasonable. It will probably change. It's evolving. But I appreciate your appreciation of it, but I think it's going to become a regulatory a fait accompli in a few years anyway.
Thank you, Paulo. Yes, we had this event at MNCT. It was a meeting to try to see if there's a possibility to make a nanotechnology study proposing some regulation. Since this bidding did not take place, they informed me that in a subsequent assessment there were no resources for that. In the general projects that do not aim necessarily at nanotechnology, we always put one of the items of the project, which are these tests. If we have the idea to propose new therapies that are less aggressive than the current ones, it is important to have these nanotechnological tests because we could go much faster in the production of new materials, but there wouldn't be any sense. If we want to reduce the adverse effects, we have to prove that the material has not this diversity. I'm from the Ministry of Health. I would like to make a consideration. This is valid to all members of the table. There are risks associated to nanomaterials that cannot be neglected. This is a consensus that was established. There are benefits, it's also out of question. The use of nanomaterials, nanotechnologies that are being developed. The main issue is how to manage the risks associated. Once we have a diversity of variables associated to nanomaterials, some more toxic, others less toxic, others more or less cumulative. We talk that sanitary and environmental authorities are not concerned with the toxicology, but I think they are, because, however, to make a regulation, we need to have the scientific evidences that are robust to establish this regulatory framework. If there is no research, there is a difficulty to have elements where the regulator can establish limits up to which point we can work. So I would like to know the opinion of this table. If there are already enough elements that can be used to establish a regulation. In the absence of regulation, it is more difficult to approve products to enter the market. So these research products that are ready are more difficultly accepted in the market. So I would like to know your opinion about what is our current situation, at which point are we already, and what could we do as rules right now. Okay, I would like to give the floor to the three components of the table, starting with Professor David, then Zulmira and Professor Anna. And would like, after the answer, I would like to close this participation because we are on time, arriving.
I agree with you that it's really necessary to determine the potential health risks, but here's the big dilemma. The big dilemma is that we have so few, in my view, substantive good data from which to make that decision. The obvious result would be to go to something faster and less expensive like high-throughput screening. However, it's my opinion that it's not ready for prime time, that many of these high-throughput screening and in vitro have not been validated for what I would compare to in vivo effects. So we have that dilemma. We're in a transition now, and it's a very difficult question, but I think we have to start doing both to see if we can find some methodologies that can be useful. Because if we just go to the high-throughput screening, we're likely to get a lot of results that have very little relevance to human health, and we'll be sorry. So it's a very good question, and it's a very tough transition.
I agree with Professor David. I also think that this is a good issue, and it's an important and very important. We have to do both things. We need much more results to have an accurate regulation, but it's important to start to think about this regulation to get oriented in that sense. Also to know which tests we should really do and which are the ones we can go faster in research. Okay.
First of all, I would like give you some my opinion. I think it's, if we're talking about nano, there is nothing nano in a toxicology assessment. We're still using the same paradigm of the technique. It's mean like it's nothing nano, by the way. Certainly we, like David mentioned it, we characterize with its different surface area reactivity assessment. But when we use cell culture, you can adjust it specific, like you mentioned, centimeter per square feet. But it's rather difficult to see this effect in vivo. I honestly do not believe much, and my colleague at NIOSH, that its in vitro study can be fully validated and translated. And I'm currently project officer for Nanomune project, which is 7 framework. And we have people from IPA who tested a bunch of nanomaterials, which is in vitro. It's not toxic, but we found some toxicity in vivo. It's mean assessment of nanomaterials should be go through the same paradigm like regular assessment toxic effect to prevent any future outcome in the humans, side outcome, side effect.
I would like to thank the participation of all in this round table. It was very rich. I would like to give the floor to Fatima to do the conclusion. I would like to thank you, to thank the speakers for the excellent speeches of this table and also the presence of all. Thank you very much.